Welcome to this e-learning program on principle involved in centrifugation. This is Ramakrishnan, Assistant Professor, Department of Biochemistry, St.Joseph's College, Cuddalore. In today's program, I am going to discuss the basic principle involved in centrifugation and the factors which governs the rate of sedimentation during centrifugation. Centrifugation is the separation technique used to separate the particle present in a mixer by using applied centrifugal field. In this case, the compound to be separated should be in a liquid medium. The force which is used for separating the compound from the mixer is called centrifugal force. That is a force which is generated because of rotation. So this centrifuge generating machine, that is centrifugal force generating machine, is called a centrifuge, and the process is called centrifugation. So this is the most widely used technique in almost all the clinical laboratories. It may be a simple one or a highly sophisticated one. All the laboratories this instrument will be present, and similarly in the research institute this will be a key role. In this case, take the simple one, that is collection of blood sample. After collecting the blood sample in a clinical laboratory, the duty of the technician is to separate the components present in the blood, that is plasma or serum, and then cellular components such as RBC, WBC and platelets, in order to analyze individually or for storage, like blood banking. Okay. When we look at the basic principle involved in this separation technique, is first one is the applied centrifugal force. The second one is size and shape of the particle what we are going to separate, and the third one is the density and viscosity of the medium what we are using for suspending the particle for separation. Now, under this, first one is the applied centrifugal force. This is a force generated because of rotation. So when we look at a complete rotation, that is 2 pi radians per second. Now we are going to use this as a omega square r. Omega is the angular velocity and r is the radial distance of the particle from the axis of rotation. That is a rotor and center part of the axis, and capital G is the centrifugal force. R is the radial distance, that is, look at this image, from the center point to the sample placing well. Now the formula for finding this one, that is omega is equal to 2 pi revolution per minute. So we are radians always represented by second. When we convert that into revolution per minute, so we are placing by 60. Now from this, centrifugal force G is equal to 4 pi square revolution per minute whole square into R divided by 3600. That is 60 into 60. Generally, this force is expressed in terms of multiples of gravitational force. That is, centrifugal force is always expressed in multiples of gravitational force. By comparing these two, that is, gravitational force is a standard one, that is G value is 9.8 meter per second square. We are going to compare the centrifugal force with the standard gravitational force. So we are using a general term, a new term called a relative centrifugal force. RCF is equal to omega square into R by G. So RCF is equal to, on further extension, 4 pi square into RPM whole square divided by 3600 into 980, that is centimeter, meter to centimeter conversion, into R. This is the general formula. On simplification of these standard values, we will get RCF is equal to 1.11 into 10 power minus 5 into revolution per minute whole square into R. that is radial distance, and then revolution per minute whole square. That means relative centrifugal force is directly proportional to the speed or the revolution per minute, the square of the revolution per minute, and directly proportional to the radial distance. That means when we increase the RPM, rate of sedimentation will be very high, and similarly when we increase the radial distance, that is the distance of the rotor point sample placing well from the center of axis. So when we change these two factors, the rate of sedimentation will be adjusted. For example, consider the simple relation. So rotor radius 6 centimeter and the machine is spinning with 5000 revolution per minute. The overall revolution, that is a relative centrifugal force generated, is equal to 1665 terms of G, that is relating with the G. Suppose when you change this 6 centimeter into 12 centimeter, we will see the doubling of the G value in relative centrifugal force. So that means directly proportional to R and square of the revolution per minute. Next, during centrifugation, the particle will experience a new kind of force. So when you apply external centrifugal force, that particles will migrate towards the bottom of the tube. So the force which acts on the particle is always expressed by 4 by 3 pi rp cube into density of the particle minus density of the medium into omega square r. Here 4 by 3 pi rp cube is the volume of sphere, and rho p, density of the particle minus density of the medium. Omega square r, once again angular velocity. Okay. Next, so during this movement, it will experience the another frictional force. F is equal to V into F. So we are introducing the Stokes formula. F is equal to 6 pi eta rp. Rp is the radius of the particle. See, R is the radial distance from the axis of rotation, whereas Rp is the radius of the sphere, that is particle separating one. So on simplification, we will find that the two forces experienced by the particle, one is upthrusting force, another one is frictional force, that is buoyancy and frictional force. So centrifugal force act on the particle, whereas the medium generate another two, one, that is buoyancy and frictional force. That means a particle is moving through the medium, the molecule present in the medium will retard the movement of the particle, that is called the frictional force and the buoyancy. So these two has to be balanced, then only the rate of sedimentation will be achieved. Otherwise the particle will float on the surface of the medium. So that will be achieved by increasing the velocity, that is F is equal to F naught. When we achieve that same, we will see the sedimentation. On equation 4 by 3 pi rp cube into density of the particle minus density of the medium into omega square r is equal to 6 pi eta rp v. These two things has to be balanced. On simplification, we see that velocity is equal to dr by dt, that is rate of sedimentation with respect to the time, is equal to 2 rp square into density of the particle minus density of the medium into omega square r divided by 9 eta. Eta is the frictional coefficient. Omega square r, angular velocity. Rho p, rho m. Rho p is the density of the particle. Once again, rho m is the density of the medium. From this, V is equal to 2 rp square, density of the particle minus density of the medium into angular velocity divided by 9 eta. So 9 eta for sphere, that means the separating molecule is sphere, means this is the formula. Suppose a particle what we are separating is aspherical. For example, spherical ribosomes, aspherical Golgi complex, like this, anything now. So if it is aspherical molecule, means that will generate another kind of frictional resistance. So we are changing that 9 eta F by F naught. That relates the frictional coefficient experienced by the spherical particle and aspherical molecule. On placing these values in this one, finally all these standard ones are all converted into a single standard coefficient S. S is called sedimentation velocity. So V is velocity, S is equal to Swedberg unit, that is S omega square r. V is equal to S omega square r. From this, S is equal to V by omega square r. Here S is the terminal velocity by unit acceleration. So sedimentation coefficient is expressed by another term called Swedberg unit. S is the sedimentation coefficient. The value is 10 power minus 13. So it is also called as Swedberg unit. 1 Swedberg unit or 1 S is equal to 10 power minus 13. This S is generally used in separating the molecules. For example, while separating the cellular components, ribosomes, 60s ribosomes, 40s ribosomes, and the S is relating with the Swedberg unit. On conclusion, so this Swedberg unit was introduced to honor the Theodor Swedberg, who has contributed a lot for inventing the ultracentrifuge, the latest advanced technique for all the micromolecules and macromolecules as well as intracellular organelle separation. But he introduced this technique for solving the colloidal particle and Brownian moments. Thank you.